In the biomolecules, the next group of biomolecules are lipids. The term lipids was derived from Greek language, in which lipos means fat. So this name was given by Bloor scientist. Here these biomolecules are the fatty acids have got esterified to alcohol or some other substances. Biomolecules, the lipids. Lipids are the fatty acids which are esterified to the alcohol molecule or some other type of organic substances. Here, these are generally insoluble in water, but when they are mixed with water, they are dispersed in the water. When they are dispersed in the water, it forms a colloidal like solution, or colloidal like nature will be exhibited by this lipids when they are mixed in the water. But these are soluble in organic solvents like alcohol, acetone, benzene, chloroform, etc. So this is the basic information related to the lipids, and as we know that these are the macromolecules or biomacromolecules which are having the molecular weight which is actually less than 1000 daltons. So as we have discussed, these are the part of biomacromolecules. But when do we say that a organic compound is a biomacromolecule, means if it have the molecular weight more than 1000 daltons, but this is the one which is having the molecular weight less than 1000 daltons. Despite of this nature, why it is considered as a biomacromolecule means, so here the lipids are present in the structural component of a plant cell, and when they are disorganized, when they are broken, so they are not independently separated out, but they are forming into vesicle like substances or vesicle like structures. Then when these vesicles are filtered, so they are retained in the acid insoluble fraction. Along with other biomacromolecules. That is the reason why these are, despite of having less molecular weight, they are the part of the acid insoluble fraction and biomacromolecules. And basically, as these are hydrocarbons, they are made up of mainly the carbon and hydrogen. Main elements which are present are carbon and hydrogen, but apart from carbon and hydrogen, they may also have oxygen, but the proportion of oxygen is very less when compared to the number of carbons present in them, number of hydrogens also. And some of them may also contains phosphate, and some they also contain nitrogen, etc. are also been seen, but this number is very present in very less number, and they are present in very few types of fatty acids or the lipids. Where does these lipids are present in a plant or any animal? So mainly these are present in a form of oils, like we know that different types of oils we will be using, like sunflower oil, rapeseed oil, safflower oil, and they are also been seen in the lycopene of a tomato. They are also been seen in the vitamins A, K, etc. So likewise, these are the lipids are present in various types of the sources which are present in different plants and also in different types of animals. So here these are basically the fatty acids. Now let us discuss what are these fatty acids and how they are organized to form into a biomolecular structures. Actually, to say, the lipids contains fatty acids and the glycerol. So basically they are majority of them having the fatty acids. Sometimes some of the lipids may have both the fatty acids and glycerols, where the fatty acids are esterified to the glycerol molecule to form a complex organization. So here in the fatty acids, as we know that these are also made up of carbon, hydrogen and oxygen. So the basic molecular formula of this one is CN, H2N and O2. So as I said that number of carbons and hydrogens will be more when compared to the number of oxygens. The number of oxygens are very less when compared to the number of hydrogens and also to the number of carbon molecules present in the particular fatty acid molecule. So here the fatty acids will exist in two forms, that is saturated and the unsaturated fatty acids. So what is the difference between the saturated and the unsaturated fatty acids? So saturated fatty acids are the one which does not have double bond in them. Such type of fatty acids are called as saturated fatty acids, whereas the one which have double bonds, so these are called as saturated fatty acids. They may be in the form of like this, C double bond C, like this. And how many such double bonds are present means it depends. So based on that one, monene, diene, triene, tetraene, like different types of unsaturated fatty acids are there. Let's discuss them after a while. Before that, let's discuss about the saturated fatty acids little amount. And here these are present both in the plants and also in the animals, both of them. So but majority of the saturated are present in the animals, majority of the unsaturated are present in the plants. So these are present in the animals. These are present mostly in the plants, and they are present in aquatic animals also. So aquatic animals which live in colder regions or cold water. So this type of variation can be seen. The plants will prepare the fatty acids by various biochemical mechanisms, whereas animals will also prepare the fatty acids with various biochemical mechanisms. But there are some fatty acids which are not being synthesized or prepared by the animals by various biochemical mechanisms. Let's see what are those. So here basically the number of carbons which are present in fatty acids are ranging from 4 to 24. The number of carbons ranging from 4 to 24, but in most of the cases, in most of them, they have the number of carbons from 16 to 18 carbons. 16 to 18 carbons. As we said that they have, along with the carbon, they have the hydrogens also. The number of hydrogens will be more than that of the number of carbons. And here in the fatty acids, here as I said that there are some animals which cannot, they cannot prepare some of the fatty acids or some of the lipid molecules. So by various biochemical mechanisms, those are oleic acid, linoleic acid, linolenic acid and arachidonic acid. So these are the fatty acids which the animals cannot prepare by various biochemical mechanisms, whereas other type of fatty acids can be prepared by the animals by various types of biochemical mechanisms. In all of them, whatever the oleic, linoleic, linolenic, arachidonic acid, so all of them having the same basic formula like CN, H2N O2. This is related to the saturated fatty acids. But here CnH2n minus 2x O2 is a molecular formula for unsaturated fatty acids. So what is this? What is the difference that we find between the saturated and unsaturated? Means here H2n is given, but H2n minus 2x is given. So here the 2x represents the number of double bonds present in the unsaturated molecule. All of these are the unsaturated fatty acids. So let's discuss the molecular formula and how they have been arranged. How many double bonds are present in each of the unsaturated fatty acids which are not been prepared or made by the animals by various biochemical mechanisms. Unsaturated one, oleic acid, which is an monine. What is the meaning of monine? Means which contains single double bond. Which contains single double bond. Actually, we know that saturated is having a molecular formula of Cn, H2n and O2. But unsaturated one Cn, H2n minus 2x O2. Here C18, H34 and O2 is present. So here as C18 are present, hydrogen should be, according to H2n formula, it should be 36, but as one double bond is present 2x, so minus 2 is there. And diene molecule, linoleic acid, where two double bonds are present. Here C18, H32 and O2, where number of hydrogens are 4 less when compared to the normal saturated compound. Triene, linolenic acid. Triene linolenic acid, which is having C18, H30 and O2. So it is having 3 double bonds. Whereas tetraene, so example is arachidonic acid. It is having 4 double bonds in it. So here number of carbons are 20. So as 4 are present, H32 and O2. So these are the examples of unsaturated fatty acids where the number of hydrogens are differing from the saturated hydrocarbons. Based on that one, these are the molecular formulas of various unsaturated fatty acids present in plant cells which are not been produced by the animal cells or animal bodies. Unsaturated fatty acids are having the low melting point. And these are mostly present in the plants and in the cold regions of aquatic animals present in the cold regions, but they are mostly absent in the other animals. And here these are having hydrophilic or polar nature and also the hydrophobic or the non-polar nature. Generally, the side which contains a carboxyl group, the side with the carboxylic group will exhibit the hydrophilic nature, whereas the other part of the hydrocarbon will exhibit the non-polar nature. And the one which is showing the polar nature or hydrophilic nature will bind with the water molecules, and that is the one which is which will form the bonds with the water molecules, and that is it will reduce the surface tension of water. And Also it is helpful for the action of water. Actually here the soaps which contains these fatty acids, the hydrocarboxylic part of the fatty acids will will act with the water molecules, because that is having the hydrophilic nature, and that shows the activity or the potentiality of the action of the soaps on the water. Then the classification of fats. The fats are categorized into simple fats, compound or conjugated one, and derived. Simple are the one which contains the fatty acid and alcohol, whereas compound are the one which contains fatty acid, alcohol, and other organic compounds in them, whereas derived are the one which contains the derivatives, other type of derived organic compounds are present in them. So this type of deviation can be seen in the classification of the fats. So next, true fats or natural fats. So here actually the fats are formed by the esterification of the glycerol with the fatty acids. So based on the number of fatty acids which are esterified to the glycerol molecule, based on that one monoglycerides, diglycerides and triglycerides are present. So let's take one example here where CH2OH, CHOH and CH2OH. This is a glycerol molecule. Otherwise trihydroxypropane, 3 hydroxyl groups with 3 carbon molecules. So we call it as trihydroxypropane with COOH, second molecule COOH, and third fatty acid COOH. So in this process here the 3 fatty acids are present. The 3 fatty acids are esterified to the hydroxyl group of only one glycerol molecule. So in this part here the water molecule is lost, H2O. Here one more water molecule is lost, H2O, and one more water molecule H2O had been lost. So it forms into a molecule that CH2O COR1 with CHO COR2 and CH2O COR3. So this is an example for triglyceride molecule. Plus 1, 2 and 3. 3 water molecules are released. Likewise, the triglyceride is formed by the esterification of 3 fatty acid molecules with 1 glycerol molecule. Whereas, based on that one, if monoglyceride had to be formed, only 1 fatty acid is esterified to the 1 glycerol molecule. If diglyceride had to be formed, 2 molecules of fatty acids are esterified to 1 molecule of glycerol molecule. Likewise, based on that one, based on the number of fatty acid molecules which are participating in the esterification process, the monoglycerides, diglycerides or triglycerides are formed. Generally, the fats, oils or the fats at normal room temperature. They are in the form of liquids, but sometimes they may become solidified when they react with the oxygen molecule. So that is called as a solidification. Otherwise, so when hydrogenation is taking place, hydrogenation of oils. So here hydrogenation of the oils or the fats will lead to the formation of hard fats So these are examples like vanaspati. Hydrogenation of the soft fats leads to the formation of hard fats like vanaspati. So likewise at room temperature, if they are existing in the normal liquid form, so we call them as oils. That is based on the melting point also, this type of variation can be observed in the soft and the hard fats or soft or hard oils. Polyunsaturated fatty acids. These are called as PUFA. So these are the one which are essential for the maintenance of health of human beings. So these are the one which will reduce the problems related to the heart. The polyunsaturated fatty acids PUFA, which contains more than one double bond in them. So polyunsaturated fatty acids are recommended by the physicians for maintenance of health of human beings, because they will lower the cholesterol in the blood and also related to the cardiovascular problems. And these are mostly been seen in the examples like oleic acid, linoleic acid, linolenic acid, and arachidonic acid. So already we had discussed about all these four. So hard fats. Hard fats are the one which are solid at room temperatures, and these are the one which contains long chain saturated fatty acids. So this type of nature will be there for the fatty acids, for the hard fats. But if you take the example of butter, butter is the one which contains short chain fatty acids. So that is the reason why it is considered to be the soft in its nature. So that is difference that we find between the butter, which is a soft fat, from that of the other hard fats. Next one is waxes. So waxes are the fatty acid esters with a long chain monohydric alcohols. So this type of lipids or organic substances are called as waxes. Generally, so waxes are a coating like structures which is present on outer surface of various plants and animals. So here in animals, various types of waxes are being secreted, so particularly by the cutaneous glands. So that is lanolin. So this is a one secreted by the cutaneous glands, and it is a water insoluble surface which is formed on animal fur. Then cerumen, so which is secreted by cutaneous glands, so this is a one which is useful for lubrication of eardrum. Then bee wax or bee waxes, it's made up of complex nature or complex fatty acids are present in them. So like palmitic acid and myricil alcohol are present in them. And other one is paraffin wax. Paraffin wax is the one which is used in making candles along with stearic acid. So it is used in the making of candles. Likewise, different types of waxes are secreted by animals and which are concerned with various economic importance also, like bee waxes and paraffin waxes, which are useful for economic purposes, whereas the lanolin and the cerumen are the one which are useful for the other metabolic activities or various other activities related to the animals only. Next one is cutin. Cutin is an outer covering, or we can say that cuticle is the outer covering which is present on the outer surface of epidermis. In majority of the plants, so except roots. Here this cuticle is made up of cutin. So this is a one which is protecting from wetting. And also from absorption. Actually here in the xerophytic plants, they are helping for reducing the rate of transpiration. Then next one is suberin. Suberin is mostly present in the matured structures like matured stems or the one which had undergone secondary growth. So suberin is present in the secondary cell wall materials. And this is the one which is responsible for impermeable nature or impermeable to water. And this ability, okay, here we know that the endodermal cells contain lignosuberin walls. Lignosuberin thickenings are present. So this, because of this suberin, so it is acting as impermeable to the movement of water from the inner cortex region to the steel region. So this type of other material is called as a suberin. So next one is phospholipids. Phospholipids are also fatty acid esters which are so in which the one of the fatty acid is replaced by phosphoric acid, and this phosphoric acid contains the nitrogen containing organic molecules. So here this is a case where if choline is present, so we call that as lecithin. If choline is present, we call it as lecithin. So if you see the molecular structure of this lecithin molecule, so here CH2O, CO, R, CH, O, R2, and here CH2 where the phosphoric acid is present. This phosphoric acid contains nitrogen containing substance, organic substance in this one, so which is CH2 and nitrogen with CH3 and 3 methyl groups. So this is choline, and this is a phosphoric acid or phosphate molecule. This type of organization is present in the phospholipid molecules, that is lecithin. And where does this lecithin is present means this is the part which is present in the cell membrane. So the trilamellar model which contains the bilayers of phospholipids, that part contains the lecithin molecule in the form of phospholipids. This lecithin is amphipathic or amphipathetic. So what is the nature of this amphipathic or amphipathetic means which contains both which exhibit both polar and also non-polar nature. So polar means because of this hydrophilic nature, and non-polar is because of hydrophobic nature. So in this one, the choline and the phosphate containing part, this part will act as a polar or hydrophilic. whereas the remaining part, so that is glycerol along with the 2 fatty acids. So this fatty acid containing part will be acting as a hydrophobic or non-polar in its nature. So here this is an organization which contains the choline, phosphoric acid, glycerol and 2 fatty acids are there. Actually we said that the lecithin is a triglyceride. So triglyceride means we should contain which is formed by esterification of 3 fatty acids with the 1 glycerol molecule. But in this structure we can see only 2 fatty acids because one of the fatty acid is replaced by the phosphoric acid which contains nitrogen containing substance called as choline molecule. That's the reason why we can see only 2 fatty acid molecules which are acting as hydrophobic or non-polar in their nature. While the choline phosphate part or the glycerol part up to here, it is acting as a polar or hydrophilic in nature. Let's see how this lecithin is organized in the trilamellar structure which is present in the plasma membrane of plant cells. Let's take the proteins which are present on this side, globulars, and here in the trilamellar structure this type of organization can be seen here, where on both the sides we can see the protein molecules. So here this side shows the water containing side. So which can bind with the water molecules like this. So this is a protein layer, this is a water containing thing. So here this is a polar or hydrophilic side of the phospholipid and this is a non-polar or hydrophobic, and one more layer of phospholipid is present. This is one layer of phospholipid, the second layer of phospholipid. In this one the polar side is towards the water. Here in this also the polar side is towards the water, so which can form the bonds, whereas the non-polar, in both of them the non-polar sides are facing towards or present towards the middle. This type of organization can be seen in the arrangement of the phospholipids in the bilayer of phospholipids present in the plasma membrane of plant cells. Steroids are the complex lipids which are having cyclopentanoperhydrophenanthrene structure. So in this one, this is an organization that we can see here which contains hexagonal ring-like structures. Where they contains the side chains on both the sides here and here. They have the methyl groups at the 10th position and also on the 13th position. So this is a molecular organization the molecular structure that can be seen in the steroids which are showing cyclopentanoperhydrophenanthrene structure. So these are the structures which are present in the animals which are helpful for synthesis of various types of hormones and also other metabolic activities. Next one is cholesterol. So here molecular structure, molecular formula is C27, H45 and OH. So this is a one which is present in animals and also in the plants. So here this is present in plants like in potato. This cholesterol is showing the molecular structure like this. OH, the double bond here and a methyl group. So this is a basic framework of structures that can be seen in the cholesterol molecule. So here wherever the corners are present, that corners represents the presence of carbons in them. So that here also we can see CH, CH3, here CH2, here CH2, here CH2, here CH, CH3 and CH3 molecule. So this is a basic structure. So wherever we can see this part here, the CH2 will be present here. CH2 likewise, where the triple 4 bonds are present here, only C will be present. Here only CH2 are present. This is a basic structure that can be seen in the cholesterol. Cholesterol have significance in the body of human beings. So here this is deposited in the arteries, so which is leading to arthrosis. And here there are two types of cholesterols are there. One is called as LDLC, low density lipoprotein cholesterol. So this is called as bad cholesterol. So when it is deposited on the surface, on the outer surface of arteries, so this will lead to thrombosis. And if it is deposited inside the arteries, atherosclerosis will be resulting. So this is a bad cholesterol which is deposited in arteries, leading to the cardiovascular problems. Then HDLC, that is high density lipoprotein cholesterol. So this is a one which is called as good cholesterol. So why it is so called as good cholesterol means, so which will removes the cholesterol from the arteries. So likewise two types of cholesterols are present, and these cholesterols are generally the water insoluble one which are present in the inside the arteries of human beings.